Hello, everybody. Welcome to this webinar. We're going to give people another minute or so to join. There are lots of people joining today, and the number keeps going up. So we'll give people an opportunity to join this webinar. So for those of you who've joined already, I'm going to share my screen. We're conducting a short single question poll. So you should be able to see a link right here and a QR code. You can actually scan this with your phone, answer the question, which basically asks you which database you most frequently use, and of course you can pick other as well. And I will also put the link in the chat window. So if you need that there, do that. Great. So I see people are already doing this. That's fantastic. So we'll keep this going for maybe another minute. I'll keep the poll open anyway throughout the webinar, and then we'll go ahead and get started. Yeah, so if you are unable to answer the poll, that's fine. You can type it in the question, but if you can, then that's wonderful. This is, obviously this is just for our information, just to get a general sense. Has no bearing on anything, so don't worry if you cannot answer the poll. There'll be more opportunities for you to tell us what databases you work on, and I will cover that as well. Okay, for those of you who are just joining, we are running a quick single question poll. As you can see on the screen, there is a QR code which you can use your phone to scan if you want, or you can type the URL that's shown up there in your browser to get to the question. This is a simple question asking you which database you most frequently use, or which you use. You can select more than one. And as you can see, we've already had over 100 participants in this poll, which is fantastic. I will keep this open throughout the webinar just so that if you have, if you want to answer it later on, that's fine. Someone is asking to move to see the poll. Okay, let me do that. I'll move the, there we go. So that's the full poll. Okay, and I'll show this again towards the end of the webinar, so we can get more. We can obviously, people who didn't get a chance to answer can answer then, and we can also see the final tally. All right, that's great. Thank you, everybody. So let's keep this open. Okay, so welcome everybody. So this is our second webinar in our series, new series of weekly webinars, which we will be holding for a while now. As you know, we've gotten many requests from folks recently wanting to get more online training, and so this is a great opportunity for us. VEuPathDB, in case you weren't aware, is a eukaryotic pathogen vector and host informatics resource. We are one of two bioinformatic resource centers that are funded by NIAID, and there's a link at the bottom of this window that has more information about the Bioinformatics Resource Center. So feel free to visit that, or obviously a quick Google search will get you to the same page. Today's webinar will specifically cover the Genome browser in VEuPathDB databases. And in particular, we're going to be talking about JBrowse. That's the Genome browser that we use. And there are a number of features that I would like to cover during this webinar. So before we get into that, my name is Omar Harb, and I'm part of the outreach team at VEuPathDB. I'm joined today by a number of outreach personnel from VEuPathDB, including Gloria Giraldo Calderon, Suzanne Warrenfelds, Sam Rund, and Evelina Bashenko. They will be taking care of answering questions throughout the webinar. So in case you weren't aware, in GoToWebinar, you can ask questions. There's a question panel that you should see in your GoToWebinar panel, and you can type questions in there, and we will monitor those and answer them either verbally, or we will answer them by typing the answer in there. You can also raise your hand if there's a question that you have, and we can try and follow up with you as best as possible. In addition, in addition to this, the webinar is also being recorded. So if you missed something during the webinar, or you have colleagues who you think would benefit from this webinar, you will get a link to this webinar, and it'll also be posted in our YouTube channel and also on the webinar page. And so I will quickly switch to the webinar page. Here. So this is the webinar page, which is linked on the slide I showed you before. Everyone who's registered will also get the link, and we also advertise it heavily in different places. You probably got an email with that link, or you saw it on social media or on our website. So you will see that today's webinar is Introduction to Genome Browsers. Next week, we're going to be covering interpreting RNA sequence data in the Genome browser. So today's webinar is a nice primer for that, for the next week's webinar. So today we'll just talk about the functionalities in JBrowse and how you can load tracks, modify tracks, explore the data in the Genome browser. Next week's webinar will be run by JBrowse, and he has a lot of information about how you can use RNA sequence data to interrogate gene models and figure out whether you trust the gene model, or if you would propose alternative gene models or splice variants and so forth. So this should be an exciting webinar. The next webinar, May 7th, will be on building search strategies. And so in VEuPathDB databases, you have a mechanism where you can combine searches into an in silico experiment, basically. So you can go in and ask, for example, find me all genes that are kinases that are also predicted to be secreted and contain a transmembrane domain, or are expressed at a particular stage in your organism of choice. So you'll hear more about this, obviously, throughout these different webinars, and if you want to learn more about building strategies, and that will be a good webinar for you. On May 14th, we will be covering the Galaxy workflows. So we have a way for you to analyze your own data using Galaxy, and then integrate the data back into VEuPathDB. So for example, you can analyze your own RNA sequence data, examine it in Galaxy, but also you can export the data back to VEuPathDB so you can run your own searches on the data. And then the last webinar so far that's listed, we will be adding many more in the coming days. But the last one listed here is manual gene annotation in Apollo. And in fact, today's webinar and next week's webinar, where you'll be exploring JBrowse a lot, are actually useful for the Apollo webinar, because Apollo, which is a genome annotation and curation platform, uses JBrowse as a key portion of the software. So this will be a good webinar to learn more about how you can be involved in gene annotation and actually make changes in a browser format. And as I mentioned, previous webinars are also posted here. So for example, if you missed the introduction to VEuPathDB resources last week, you can click on this and go down to this section, and you'll see there's a link to our YouTube channel. Okay, so as I mentioned, today we'll be covering the Genome browser. So quickly, VEuPathDB is a merged resource between the VEuPathDB resources, which many of you from the poll would be familiar with, and VectorBase, which also a number of you on the poll as well who would be familiar with that resource. And so this is actually a really great opportunity for the Bioinformatics Resource Center to put us together so that we can actually bring together the best of both worlds. And in fact, we're very excited about the things that we're working on under the hood, and hopefully we'll be able to provide them to you so you can start accessing our resources in a unified front and a unified manner. So this is very exciting and should be coming to you hopefully in the not too distant future. And I think many of you obviously are familiar with VectorBase. So today I'm going to be focusing on using one of our resources, ToxoDB, but all of our resources function in the same way. And as we merge and release our merged resource across all of our organisms, including vectors, they will all look the same, they will all function the same, and will all contain the same tools and functionality. So the Genome browser is a way for you to browse the genomes and load various types of data in the Genome browser that are mapped to the genome, and so you can explore that kind of data. And to access the Genome browser, there are different ways of doing that. One is on the home page, there's a tools section, and there's a link to the Genome browser in the tools section. Or from the menu at the top, you'll see here there are different options. One of them is the Genome browser. And if you click on these, it's going to just take you to a default area in the Genome browser in this case, of Toxoplasma. So we have to select a default genome and a default gene for you to look at. The other way of getting to the Genome browser is if you are on a record page, like a gene page, for example. And there you are specifically exploring a gene. So for example, if I jump to this gene here, so I just picked some hypothetical gene on chromosome 7b for Toxoplasma in this case. And you'll notice on the gene page, there are different sections. And some of these sections have embedded browser views. And so for example, here where there's a gene model, so this is the gene I'm looking at, you'll notice that there are some tracks loaded here that tell you something about the gene model, the confidence in the gene model, and whether there are potential alternative gene models. But this is a genome browser view. And if you want to explore this further, you can click on View in JBrowse, Genome browser, and this will take you then to the Genome browser with those tracks loaded. One thing before I forget, the Genome browser, or JBrowse, is not developed by us. So just to be clear, this is developed by the GMOD consortium. They develop many amazing tools and software. So we're very fortunate that we can actually take advantage of what they've developed in JBrowse. And JBrowse is actively being further developed. And so we're very excited about the next versions or next generation of the Genome browser, or JBrowse, which we hope to make available to you, of course, as soon as there's a stable version that we can use. So, so I'm going to take you to this example from the gene page, and I'm just going to click on View in JBrowse, and I'm going to go using this gene model from the gene model section. And so once you click on this, it's going to load essentially an embedded genome browser view for you right here. And so depending on how much data tracks are being loaded, obviously this sometimes takes a little bit more or less time. And as you can see, I'm looking at the same exact location that was on the gene page. So this is my gene of interest. These were the two genes that were in my view on the gene page. And there are a couple of tracks that are loaded down here, which were also visible to you on the gene page. It's possible that you don't really care about these tracks and you're interested in other tracks, and that's totally fine. And so you can easily just close these by clicking on this little X at the left of the name of each of the tracks. Okay, and we'll explore this further. So the Genome browser can be divided up into three general sections. You have the menu bar at the top here, and we'll explore the different aspects of the menu. There's the location bar right here, which allows you to move around, change your location, or it's called a navigation bar technically. And the central portion here is the genome section. So this is where you will be viewing elements mapped to the genome, whether they're gene models, RNA sequence data, synteny tracks, mapped peptides. There are many different types of data, which we'll explore some of that in a few seconds. So the menu bar, each of these names up here, so genome, track, view, and help, if you click on these, they will offer you additional options. So for example, under genome, this is a way where I can quickly toggle between the genomes that are in ToxoDB. So for example, if I came to this page and I'm doing some work and I've decided, you know what, I really want to switch to another genome because I want to find out if there's some element or feature on that genome that I'm interested in as well. And so you can quickly toggle between genomes right here. The track menu item allows you to open additional track files, and I will show you how to do that in a few minutes. There's something called combination tracks. We will not cover that in today's webinar. There are various reasons for that, but suffice it to say, it won't be terribly useful for our discussion today. But it does allow you to combine different tracks together, and so if you want to view them layered on top of each other, that's potentially a nice option. The add sequence search track is very nice because it allows you to find a particular sequence in your genome that you're viewing right now. And again, we will demo this as well at some point during the webinar. View, so again, it gives you additional options, and we'll demo these. So for example, you can resize tracks. So if I'm viewing 10 different tracks in my browser, I can resize them all in one shot. So I don't have to resize each of them. And this will become more clear to you as we explore these further. And help just gives you more information. So if you want to know more about this Genome browser, it tells you we're using JBrowse version 1.16.3. I think right now it goes up to maybe 0.9 or something. And you can, if you're curious to know, well, why, what is in the newer version, this is all obviously available on the JBrowse website. All right, the navigation section right here in the middle, this allows you to move around in your browser. So for example, I can use these minus and plus icons right here. These are zoom in and zoom out icons. And you'll notice there's a big one and a small one. The big one zooms out more. So if I do this, you're going to zoom out and load more genes in your view. If I can zoom back in by clicking on the big plus sign, okay? And if I want to zoom out not as much, right, I can click on the minus sign, and so now this gives you a smaller zoom out window and a smaller zoom in window. I can move around by using these arrows, so you can click on the right arrow, and that's going to move you a certain distance to the right, or I can click on the left arrow, and it's going to move you a certain distance to the left. I can also use this menu item right here to switch between chromosomes or supercontigs or scaffolds or contigs that are available and loaded. And so, for example, in this case for toxoplasma, the current version of the genome has 14 chromosomes which are loaded. And so you can easily toggle between the different chromosomes as you work on a particular genome. You can also switch to specific contigs that are potentially part of the assembly or are not assembled in there. So that's potentially of interest and utility. And then this window here allows you to type in a specific coordinate or a specific ID or even a specific name. And I will demonstrate this right now. I'm just going to start typing the word cedoheptulose. I'm looking for a gene called cedoheptulose 1,7-bisphosphatase. And you'll notice that it auto-completed. And now I can select this gene to go and visit that particular gene. So once you click on it, now it takes me to that gene page or that gene view. And this is the gene that we're interested in. And if you mouse over it, so I'll just have my mouse over it, you can confirm that, oh yeah, this is the cedoheptulose 1,7-bisphosphatase. You can also enter coordinates. So if you have a specific location on the genome that you want to go to, as long as you follow the format that's listed here, you'll be able to get to that location. There are a couple of other options here in the navigation bar. One of them is a highlight option. And I'll show you how this comes in handy in a bit, but I can click on highlight and I can say I want to highlight only this region here, okay? And so this basically anchors a highlighting point here, but you can use this for other aspects of your browsing, which I will demonstrate in a minute. And you can cancel the highlighting by clicking on the highlight button again. And then this icon here allows you to hide the track names. And so if these are getting in your way and you don't want to see them anymore, you can just simply click on this and that hides the track names, or you can display them again. So all of these are really ways for you to get around, configure your view a little bit better, and adds also some functionality. So there are a couple of other ways that you can zoom in or zoom out, right? So up here, I can, if I want to zoom further out, but I don't want to click on the plus or minus signs, I can just click on this area and select a particular region. So I'm clicking and dragging. And then as you do that, it takes you, obviously I clicked and dragged it, and I moved at the same time. So we moved, as you noticed, but also it zoomed that particular area. So now we're looking at this entire area right here, shown in this red box, and it contains all of these genes, okay? But I could also, in my genome view, I can zoom in as well by clicking and dragging. So I can click right here in this where the numbers are and drag. Let's say I want to just zoom into this particular region, and so now it's going to zoom into that region. And I can continue doing this until I'm satisfied with the zoom area that I want, basically. Okay. So let's go back to our
Omar.
Yes.
There was an attendee who had their hand raised, but I'm afraid that trying to find who that was, I might have deleted it. Was it Anastasia?
Okay. So if you have a question, I would suggest that go ahead and type the question in the question poll, in the question tab. And so if for some reason you're still not able to type the question, raise your hand again, and I can obviously unmute you, or we can unmute you and you can enter your question or ask your
Excellent.
question.
That's great. Thank you. Yeah.
Mm-hmm. Good. Thanks, Suzanne. Okay, so I'm going to go back to our set of heptulose gene. Okay, so here we are back in this area. And so this is our gene. So you'll notice that, so one more thing that I did not mention is to move around, if I click and keep my finger on the mouse, right? So I'm clicked down in the genome view section, then I can move around. Okay, so I can basically, it's almost like you're on a map in Google Maps, so I can just go back and forth as I wish. So if for example, I'm looking here in the center, but I really want to see what's the upstream gene, so I can go here and see that there's a hypothetical protein up here, and then I can move down a little bit and see what's on the downstream side of this, and here's another gene, it's another hypothetical gene, okay? You'll notice that some genes are red and some genes are blue. And so those denote the orientation of the gene, whether they're on the forward or reverse strand. And you'll see that there's a little arrow at the end of this, at the sequence, that indicates the direction of the gene. Okay. So the other thing you can do with these tracks is there's a lot of information available to you actually on the track itself. So before we actually load additional tracks, there are a number of things you can do here. So for example, this gene, when you mouse over it, you get a little pop-up here that shows that this is a set of heptulose 1,7 bisphosphatase. But if I click on it, I get additional information. So this tells me that this is in the ME49 strain of Toxoplasma gondii. It gives me the gene ID. It tells me that it's protein coding. It tells me the name, the description of the gene. It gives me the exact coordinates of the UTRs, which are shown in gray and in the background. So 5' and 3' UTRs. The 5' UTR is at the top here. And then you'll see the coding sequence. So it tells you the exact location of the coding sequence, and then the 3' UTR. You can download the coding sequence or the protein sequence of this particular gene. You can go to the orthology group of this gene. So if this gene has homologs in various organisms loaded in ToxoDB, you can access them right here. And then you can quickly go back to the JBrowse view or to the gene page itself. So let's say you come across this gene while you're browsing the genome, and you say, I wonder what else I can find out about this gene. I'm going to go visit the gene page. I can click on this link right here, and it's going to open the gene page for me. So the other place you can interact with this track is by clicking on About this track. And so depending on the track, you may get more or less information, but this just gives you a general information about what this track contains. And then there are additional things that you can do right here, which some of them are going to be, you might find very useful. Some of them are not that useful. So it really depends on what you're doing. For example, you can change the view. You want it normal, compact, or collapsed. And so this will preserve space for you depending on what you're doing. You can save the track data. And I will demonstrate this by showing you how you can save the sequence of a particular region of interest. You can delete the track, obviously, from your view. It doesn't delete it forever. You can pin it to the top. So if you pin a track to the top, that's going to stay at the top of your view. So I can load many tracks on the bottom here, and this will stay pinned to the top. And this is a useful feature when you're loading many tracks and you want to keep looking at the gene model sort of right in front as you're scrolling through different tracks. And we'll demo this later on. So one more feature that you can do is, again, I moused over the gene model and I right-clicked on the gene model. So you can do this by either if you're on your on a laptop and you don't have a mouse with you, you can either click on Control click, or depending on your operating system, you may have different key combination. Or if you have a mouse, you can right-click on that. And then this gives you additional information and functionality. So I can view details. Again, this is the same stuff that we saw by just clicking on the gene. So that was the default. I can go to the gene page, or I can highlight the gene. If I click on highlight gene, it's going to just highlight that exact region of the gene. Okay, so you don't have to do it manually if you just want to highlight that gene. Okay, so pretty much every track that we're going to be loading today has various things you can do with it, and we'll try and demo some of the more frequently used ones. So I'm looking at one track here, which is the gene models, but let's say now I'm interested in loading additional tracks. So any data that we have in the database that is mappable to the genome, in general, most of the data we load it in the Genome browser. So you have access to it, so you can browse it as we're looking right now. And so to explore what data sets we have loaded in the Genome browser, you can click on the Select Tracks tab, which is up on the left side of your Genome browser, in JBrowse. And when you click on this, you will notice here that there are many tracks that show up. So it tells you here there are 496 tracks, right? So that's a lot of tracks, and it's obviously going to be very hard for you to just find the track you're interested in if you're just going to start scrolling through these. So we try and make this easier for you in two different ways. One is you can start searching for a particular track. So if you know exactly what you're looking for, you can start typing words here, and then it'll filter the results for you. Or you can start using these categories here on the left to narrow down what you're interested in. So for example, if I'm interested in looking at proteomics data, I can see here there's a category called proteomics, and then there are 21. So there must be 21 tracks for proteomics data. So if I click on this, this is going to filter all my tracks right here. You'll notice right away that you have this little red X that shows up that allows you to clear filters. And that's, of course, if you're switching between filters and you start searching, it's searching in your filtered results. So if you want to go back and search everything, you're going to have to clear your filters always. But I clicked on proteomics, and now I have my 21 proteomics tracks that are available. And this may be sufficient for me to find a track of interest, or you may need to subcategorize these even further. And you'll notice, because sometimes a data set may have more than one track, so depending on the type of experiment. And you'll notice here on the left, under the category, there's a subcategory. So in this case, it's just protein expression, so nothing too fancy here. And then you'll notice here there's a list of data sets that meet this criteria. And then there are additional tracks down here, and those become more useful for other types of data as we look at these. But I can take one of these tracks, and if I'm interested in loading any of these tracks, I can just start clicking on them. So let's say I'm interested in this proteome during human cell infection. I can click on this. I can select maybe this extracellular vesicle one, mitochondrial matrix one, And again, these are peptides, so who knows if they're mapping, but as I'm clicking these, they're actually loading in the background. And so I can quickly go back and see what this looks like. So I'm gonna click on this, either I can click on Select Tracks tab again here on the right-hand side, or I can click on Back to Browser here. And this will take me back to the browser. And you'll notice that there are some peptides that loaded. So now I have additional tracks that have loaded. And some of these tracks, are, depending on the track you load, have a lot of information in them. So you can see here this peptide that mapped to this exon, or the part of the exon that translates to this peptide, came from, if you mouse over, it gives you more information, came from the 44-hour infection time point. And then you'll see here's another experiment from the extracellular vesicles. In this case, this peptide was mapped to open reading frames, so we're able to actually tell you that this peptide comes from 2 different exons, and so this is quite nice. And the same is the case for this particular experiment. And so you can use this information to give you support that this intron is actually real, at least in the translation product. Okay. And once again, depending on what you're doing, you may be interested in modifying additional aspects of the track, but again, it depends on the type of track. You may have more or less options available to you. I'm just going to click on one of these tracks. So let me go back and show this again. So I'm clicking on this little arrow where the name of the track is available here. And now I can click on About this track if you want to get more information about this track. And you'll see here that there's a short description about this experiment. And then there's a link to a dataset page. And so this will take you to a dataset page where there's more description about the proteomics data. And so you can explore this and find more information about this particular dataset. Okay, so I'm going to go back to my track selection, and I'm going to go ahead and uncheck these tracks. I don't want to look at them anymore. I'm going to clear my filters as well. And now let's go ahead and take a look at the sequence. So sometimes, or often we get a question, how can I view the actual sequence? I want to look at the sequence. And so under sequence analysis, and again, you can try and either find it by clicking on some of these and seeing what information is available to you, or you can start typing up here. And currently the sequence is called reference sequence. So if I just start typing reference up here, you'll notice that the reference sequence appears. And so it doesn't really matter which category is under. I could have cleared all my filters and just started typing reference, and then it'll filter down to my reference sequence. And so this is the genome I'm looking at right now. In this case, it was Toxoplasma gondii ME49. And when I click on this and go back to my browser, what you'll notice right away, it tells you that, oh, zoom in to see sequence, right? So we're zoomed too far out for this to make any sense, obviously. And so so we'd have to zoom in a bit more. And you can click on the plus sign until the sequence starts appearing. The first thing that will appear with the sequence is, now we're still zoomed out too far. So if the letters were actually written here, you won't be able to read them. They'll be too small. But this color code tells you something about what amino acids are represented there. And again, if you look at about this track, or actually somewhere in here, there's information about this. So this is, oh yeah, so I'll show you one more hidden feature here, which I would, probably nobody will use this, but just so you know, if you click on, I use this sometimes when I'm just not sure exactly how this track was configured. And if you click on this edit config option, this will open up a window where you can actually edit this. Now, don't worry, you can almost never have to edit this. It looks a little bit scary, but it's actually, if you look at this, you'll find out that there's some information here that can be parsed by a human. I'm not a computer scientist by training, so for me, I often have to take a while to look at this and understand what it says. But there's some things here that are quite obvious. But in this case, it tells me the protein color scheme is a Taylor's color scheme. And if you look this up, you'll find out that there's a color scheme, and each amino acid is represented by a different color and so forth. You can actually start modifying this if you want. and do some changes here. But sometimes, depending on if JBrowse accepts those changes, it may or may not work. And so typically you would not have to do this. I typically use this configuration track if I'm trying to pull out more information that's just not readily available anywhere else. It's usually sitting in the configuration track. Okay, so I'm gonna zoom in a little bit more. And if you continue zooming, you'll notice now that you start seeing your nucleotide sequence and your amino acid sequence in a 6-frame translation. Okay. So let's go ahead and zoom out a little bit more. I'm going to zoom back out to the size of the gene. So let's say I'm now interested in downloading some sequence. Okay. And so as I mentioned before, each of these tracks, anything in these tracks can be downloaded or can be saved. And you can see that the gene models can be saved, right? But it's kind of counterintuitive, but the gene models actually don't contain the actual sequence. They're simply landmarks on the genome, right, on the reference sequence. So the reference sequence is actually what contains your sequence. That's what we were able to see when we zoomed in, we saw the sequence. So let's say I'm interested in getting the sequence that's between these two genes right here, okay? So what I'm going to do is I'm going to click on highlight, okay? And I'm going to highlight this region between the two genes. So now it's yellow. And now I'm going to go to the reference sequence. That's the track that contains my sequence. I'm going to click on the arrow and I'm going to select Save Track Data. So now once you do this, you'll notice that you can save different things. I can save the highlighted region, which is what I want, or I can save the visible region. So let's say I want to save everything that's in my view. You can choose visible region. And then I can select FASTA, which is the actual file format. It's just a text file, basically, that contains the sequence. And so now I can take a look at this, and either I can save it, it'll save it on my computer, or I can view it. And if you click on view, it's going to produce the FASTA file for that particular region. And so this sequence is exactly the sequence that falls in between those two genes. You get also the location on the chromosome, and also so the chromosome number, and also the coordinates on the chromosome. Okay, so this is actually quite a high, frequently requested option, and this is how you do it. So hopefully that was useful. All right, so let's go ahead. I'm going to close the reference sequence track, and I'm going to switch now, and let's look at some RNA sequence data. And so I cleared all my filters. Now I'm going to go down and look for RNA sequence data, and you would expect that RNA sequence data would be under the transcriptomics category. So I'm going to select transcriptomics, and you'll notice here, oh yeah, there are 242 tracks for RNA sequence data. So I can click on this, and now as you scroll down, you'll notice that there are different data sets. And so this is nice because you can quickly scroll through these and determine which experiment you're interested in. So let's say I'm interested in this feline enterocyte experiment. You'll notice here it has a number 31, so there are 31 tracks associated with this experiment. So once I click on this, now everything that's showing up here in the middle section are all the tracks that are associated with this experiment, and there are many, right? And you may not necessarily want to look at all of them. And you'll notice here that the column headers have some information that could be quite useful to you. Some of it is obviously obvious. This is a track type. They're all coverage tracks. A subcategory, they're all RNA sequence. They all fall under transcriptomics, so nothing too surprising here. The name of the track is quite useful because this tells you something about the samples that you may want to look at. And also these alignment tracks here is also some useful information. So you'll notice that there are non-unique and unique aligners. And so we align all the sequence reads from an RNA sequence experiment. And if there are some reads that align non-uniquely, so they align to more than one location because they're repeat regions, we don't throw them out. We actually generate a track that shows you the non-unique aligners. And that's quite useful because that will tell you something about whether a gene has repeat regions and whether you can trust the expression values from that gene, because we don't use those to actually calculate levels of expression. And the unique ones are the unique ones. So those are potentially the ones that you want to look at first. And in this case, you'll notice that this experiment has forward and reverse. So this is a strand-specific experiment. So that's pretty cool. So you have reads that are coming from the forward and the reverse strand because it was a stranded RNA sequence experiment. And so this would be quite useful to look at. So let's go ahead and select some of these samples. I'm going to look at the unique from a couple of them. So unique forward, unique reverse from this first time point, the anti-Ursite S1 time point. And you might notice already in the background here, some of these tracks are loading up. And so I can also select, let's pick, I selected non-unique, so I'm gonna scroll down. Let's pick the ES2 unique. I think that's probably enough for now. And I'll go ahead and go back to the browser. And you'll notice now right away that we have our gene models are still up here, and we have the tracks for forward and reverse data, and we'll explore these a little bit more. I'm going to go ahead and pin this to the top. Okay, so now you can see how pinning to the top is quite useful because my gene models stay up here. They don't scroll off the window, but I can now scroll within the bottom window here so I can view the data and still see my gene models in view. All right, I'm going to zoom out a little bit here so we can explore this data a bit further. And so, as I mentioned, this is strand-specific data. And so we have forward reads and reverse reads. And so, and you'll notice that the colors match the gene models, right? So this gene right here is a blue gene, and you'll see that the reads are in blue. So those are on the strand that matches this gene model, or let's call, in this case, they're called the forward reads. And then if I go down here, you'll notice this gene that was upstream is going in the reverse orientation. It's a red gene, and so the reads are red as well. Okay, so that's the matchup here. Okay, remember for the tracks, I could, in the view section, I can apply some global parameters on certain tracks. And so, for example, I can set, let's just say this one, so for anything that's quantitative, so these are quantitative tracks, I can set the size, so resize it. So this is just by pixel. So this is just making it bigger. So I can go up to, let's say, 150, hit OK, and it's resizing all my tracks, right? So now they're bigger. So if you just want to see them a bit better, you can resize that. The other thing I can do is I can set the Y max, okay? And this is for linear. Right now we're looking at log data, so I can actually, let's do that. So if I do this now, what happened is I'm back to a linear scale. So now we're looking at the actual read numbers. And then again, under view, okay, we're going to set the y-axis, and let's change this to 100 reads in this case. And so now you'll see that my y-axis is changing as I'm changing this, and it's doing this globally for all my tracks. Okay, so this is a nice feature if you loaded all the tracks from one experiment and you want to quickly view them all with the same y-axis. You could obviously do this for each track independently. So let's say you only have one track, or for some reason for one of them, you really want to change the score. Let's go back here. Yeah, so let's say for, I don't know, for this track, you want to make it into a log scale for whatever reason, you can do this, and now all of a sudden this track is on the log scale. So it's important always, obviously, with any experiment, whether you're reading a paper or looking at a genome browser, to make sure that the axes all match up and they make sense. Okay. So as far as being able to modify the tracks, so another type of track that I'm going to show you, which has a lot of features. And so just as a reminder, next week, the RNA sequence analysis in the genome browser webinar will actually really focus on these tracks. So there'll be you'll have, if you join the webinar next week, you will get a lot of experience and instruction on how to modify these tracks, how to use them to explore gene models, and there are many other tracks that you can use to do so. And so I would encourage you to sign up for next week's webinar and to learn more about how to explore RNA sequence data in your experiments. So I'm going to go ahead and clear all my filters. Let me see the currently active tracks. So I'm going to, I want to keep the annotated transcripts track, so I still want to see my annotation, but I want to get rid of all the RNA sequence ones. I'm not interested in them anymore right now. So we can go ahead and close these. I should just do all at once and then select these. There we go. And so now, as you can see, I'm back to my original views, my gene models. So the other track that is quite useful and often interesting to look at is a track that's allows you to compare genomes that are in the database. So there are many different genomes loaded in ToxoDB, for example, as I showed you earlier when you looked at the list of genomes that are available. And those genomes can be selected or can be compared to each other. And so if you look under comparative genomics, let's see if I can click on this, you'll notice here you have a track called synthetic sequences. And let me go ahead and just turn this on and then we can take a look at it. I'm going to go back to our browser. So this track actually loads a lot of data. So it's loading all the gene models from the different genomes that are loaded in ToxoDB in this case. They're aligned to each other, and the shading then indicates regions of homology between them. So this tells you two things. One thing tells you something about the collinearity of these genomes. So how collinear they are the genes in the same order between the different genomes? And are the genes or the sequences in this region, are they orthologous to each other? Are they homologous to each other? So the shading will indicate that. So sometimes if a region has some kind of a transversion or something weird happening there, you could see the orthology is crisscrossing or doing something weird, and that usually indicates or something for you to investigate a bit further. So having obviously every single genome that's in the database loaded here may not be terribly useful, right? So from your perspective, the question is, well, what organisms do you want to compare to each other? And so the cool thing about this track, so this is a single track with multiple subtracks, basically. So I can click on this little arrow, and you will notice here you can select subtracks. So if I select this option, now I can go in here and choose which organisms I want to view aligned to each other. And so I can go ahead and just select clear all, basically. And now let's just go in and cherry pick a few of these. So I want to pick Toxoplasma gondii MA49, okay? And now let's say I want to select Neospora or Neospora. And so let's go ahead and just type Neospora. And again, remember, I'm only searching in my filtered results. And so this is quite nice because I quickly just filtered the results I'm looking at. Here's Neospora. I'm loading the genes and the actual genome expands. Let's go ahead and pick Sarcocystis. So if I just start typing, you'll notice here there are two Sarcocystis neurona genomes. So I'll select, again, one of them. And let's go ahead and pick Hamondia, for example, double M. And so it shows up right here. Okay, and you can keep going and choosing genomes that you think would be interesting to compare to each other. And then when you're done doing all of this, you have to scroll down to the bottom and click on Save. Okay, this is an important feature of the subtracks, is that you have to select the Save option. So once you click on Save, it now reconfigures your track with all of these subtracks displayed. And again, now I can zoom further out or further in, and then you can look at larger regions and compare synteny within those regions and of course move around. This is loading a lot of data, so the more data you load here, obviously there's more that's going back and forth between your web browser on your computer and the server, so sometimes it takes a few seconds for it to load, but I don't think it's terribly bad. Okay, so the other thing you can do, so let's say you're looking at this region, and that's something I forgot to mention right in the beginning, but if you're interested in this region and a colleague of yours is also interested in the region and you wanna share this with them and so they can explore it further, you can click on this share icon in the upper right-hand corner. And if you click on this, this shows you a URL that you can copy and paste to a colleague of yours. And when they click on it, it's gonna open up this exact view. And so they'll see exactly what you were looking at. So that's quite a nice, useful feature for collaborative work. Okay, so the final thing I think I'm going to show you in this webinar is the ability to load your own tracks. And so often, if you are generating your own RNA sequence data and you have coverage plots, or you can also generate, there are many different types of files that the Genome browser can accept. And so there are a lot of things you can do. And so actually there are two things I'm going to show you. I'm going to show you how to load a track, and also I'm going to show you this sequence search because it's a frequent question. And so I'm going to go ahead and load a track. So I'm going to click on Open Tracks, and you'll just get this window here. And already I have a couple of bigwig files. These are a result of an RNA sequence analysis. which I'm going to just drag and drop in this window. And you'll notice right away it understood that they're BigWig files. It's looking, they have to be the right format. So the first few times I tried doing this, I actually made mistakes and I had to figure out what the mistake is. But luckily, Google is very helpful, but also you can always contact us. So if you click on the Contact Us link, which is at the top of the page, we're more than happy to answer your questions. In fact, we really love hearing from our users. So please don't hesitate to use that for any question you have. And so once I load the tracks, you'll notice here it understood the file formats. It tells you the new tracks that it's going to load. And now I can click on Open. And then as soon as I do this, it loads the tracks and it shows them for me. So this is an experiment that I was playing around with. And so now I can look at this in the context of everything else that's in the genome. And this is private, right? So this is not visible to anybody else. It's being fed straight from my computer. And you can also configure this to actually accept not only files that are on your computer, but let's say you have the files somewhere on a server, you can actually get them from there. As long as there's no password involved or anything, then you can get them from there as well. And the final feature I wanted to show you is this add sequence search track. And so this allows you to actually look for a particular string of nucleotides or amino acids. And you can also treat this as a regular expression. The nice thing about this, if you're not familiar with regular expressions, they're actually quite useful. We have a dedicated search to regular expressions in ToxoDB, and we might have a short webinar just talking about regular expressions, but we also have a number of tutorials available to you that you can use and play around with those. They're not that hard to use, and they're quite useful because it allows you to put in an ambiguous sequence. Let's say you want to find a couple of amino acids followed by a proline, followed by any amino acid, followed by 2 hydrophobic ones, and so forth. You can build a regular expression like this. But also you can just type in a sequence. So let's say you're interested in finding a particular motif in your sequence. And so I can just type, and now I'm just typing something random. So who knows if this will find anything. So let's try this. And if you click on search, of course, now it tells you you're too large to view in. And so that's fine. Let's go ahead and close a couple of these tracks. We don't need them anymore. And then you can start zooming in until you are in a zoom level that you can visualize. And sure enough, it found a few of these. So you'll notice it found one here. So there's this sequence. And if I click on it, it tells you exactly it was 9 nucleotides long, which is true. That's what I put in there. And it tells you the exact location where it found this. And so that's kind of a nice way to quickly look in a particular region for a specific sequence. You can also use it to type in the restriction site, for example. So we get a frequent question: do you have a tool to find restriction enzymes in the browser? And the answer to that is this string allows you to type in the actual palindrome of the restriction site, and then it'll find it. And so that's a very nice way for you to find where the enzymes are. And you can create as many of these tracks as you want. So you can have a track for each enzyme that you're interested in if you're, for example, trying to clone something. Okay, I think I covered everything I wanted to cover today. I know this was a lot, obviously, and there are probably many other hidden features that we didn't have time to touch on. But I will definitely, as I mentioned before, this webinar was recorded or is being recorded and will be available to you afterwards. So feel free to watch it again or share it with colleagues and send us additional questions. I'm going to go back to our intro slide here. So again, all of our webinars are posted on this website, which you will get a link for if you don't have it yet. And also you will see it in emails and posted on our website. And as I mentioned before, in case you missed the beginning, the next series of webinars that are scheduled next week is interpreting RNA sequence data in the Genome browser. So there'll be a lot of Genome browser work, but really looking at gene models and trying to figure out whether you trust the gene model based on the available data that's loaded for you, or whether not only trust it, but also can you come up with potential alternative transcripts that you may want to actually explore. And that's very important. Or are you trying to define 5' and 3' UTRs of a gene, which if they're not part of the annotation, you can use the RNA sequence data to explore that further. there are other types of data sets that are available, including ChIP-seq and ChIP-chip data that's loaded, and those will be explored as well next week. And then I won't go through all the rest of the webinars for the rest of the for the next 5 weeks, but they're all listed here. And you will also get a survey after this webinar. And kindly, I'm asking everybody to take a few moments and answer that for 2 reasons. Obviously, we'd love to hear feedback from you, but also to let us know about what other webinars you might be interested in learning more about. And so if you're interested in doing more more genome browser work, we can schedule another webinar that focuses on a specific aspect of the genome browser. Or if you're interested in exploring the RNA sequence queries that are available in the databases, we'll definitely schedule one of those. Those will be on the schedule, but if you think that's really important, obviously we'd love to hear from you. And I think with that, I'm gonna give a few minutes for any last-minute questions. Maybe any of my co-moderators on the call. Are there any other questions that we need to answer verbally, or things that you're still answering?
I think we have one question where someone is asking, can you order can the order of subtracts to be reconfigured?
Right. So that's a good point, which I forgot to mention. And so let's go ahead to go back to the Genome browser right here. And so any track, so let's go ahead and load a few extra tracks here. So we have, so here are the tracks that I had on before. Okay, so now I'm back to having, 3 or 4 tracks loaded. And so what you can do easily is with any of these tracks, I can click on the name of the track and grab it and move it. And as you can see, it turns green when it allows you to move it there or down here. So now they're flipped, right? So that allows you to move any of the tracks around. And so you can order them as you wish. And so let's say, you really want this search track to be all the way at the bottom, you can move it. Sometimes you have to drag it a couple of times just because of the size of the window. So you have to be in the field and then you can move it. Okay.
Omar?
Mm-hmm.
We had a request to see the final polling results.
Oh, good point. So here we are with the final polling results. We had 131 participants who voted, and I can actually put up the, let's do this here, get out from here, and I'll move this aside as well. So if you haven't answered the poll yet, so the poll was a simple poll asking you if you which database you use, and you can scan this QR code with your phone or type the URL in your browser. Let me go back here. I'll show the link again in a second. I'm just going to make this a little bit smaller so you can. Okay, so now you can see both. So, and I can see some people are answering some more. So I'll leave this up and answer any other questions that may arise. So, and again, as we wrap up the webinar, thank you again very much for joining. We had, I think, close to 220 people or so who joined the webinar, which is quite exciting. And as usual, again, and I can't stress this too much. If you have any questions or any concerns or any issues that you come across in the database or requests, if you have new data you want to propose or new functionality, just click on the Contact Us link on any of our websites, from Vector-based to FungiDB to MicroSpiriDB, and just let us know about these features. Tell us what you can't find. If you're trying to find something and you're just not finding it, don't assume that it's your problem. It's actually, if you can't find it, it's actually our problem, and we'd love to hear about that. That tells us something about how people are searching our databases. But also, you know, you don't want to be too frustrated. So if we can give you a quick answer to help you, that's obviously would be much better than you being frustrated for a while looking for something. So definitely send us an email anytime you have a question, and no question is small enough. So we are, or too small, we're always happy to answer these questions. Okay, with that, I thank you all for attending, and hopefully we'll see you next week. Thank you very much, and everybody stay healthy. All right, bye-bye.